Welcome viewer, welcome to our Glory Globe TV. Viewers, a new study challenges a long-standing assumptions about how cyanobacteria survive environmental stress. The study led by researchers at the Israel Oceanographic and Limnological Research, the Canarite Limnological Institute shows that survival under prolonged heat stress is not determined solely by the ability to protect photosynthesis. Instead, survival may depend on a remarkable shift in cellular energy balance, with drug respiration compensating when photosynthetic electron transport becomes impaired. Microcystis aeruginosa is a toxic cyanobacterium that forms harmful algal blooms affecting water quality, ecosystem health, recreation, fisheries, and drinking water resources worldwide. Globally, microcystis blooms are usually associated with warm water conditions and are expected to intensify as climate change drives rising temperatures in Lake Gennesaret, Sea of Galilee. However, the local strain of microcystis shows an unusual seasonal pattern. This published in Science Advances, the study compared two strains of Microcystis aeruginosa, a toxic cyanobacterium that forms harmful algal blooms in freshwater ecosystems worldwide. Viewers, one strain originated from Lake Gennesaret, where Microcystis displays unusual behavior. It repeatedly blooms during late winter under relatively cool water temperatures. So the local strain was compared with the frequent studied strain PCC 7806. Viewers, to examine possible explanations for this phenomenon, the researchers used a unique experimental approach. First, they induced heat stress using an extreme temperature increase of 20 degrees Celsius, an approach commonly used in photosynthesis research but uncommon in ecological studies. Second, they waited 48 hours after inducing heat stress to assess how each strain responded and attempted to survive the heat shock. In other words, they were examining how the cells regulated their energy budget through photosynthesis and respiration in severe heat stress. BNS exclusive report.